Looks like we're still waiting on a couple more, right, Jay?
All right, let's just get a couple more people get here and then we can get started.
Okay. Yeah, we'll give it a couple minutes.
I guess before we get started, I definitely see some familiar faces there in the audience or names.
Yep. Everyone who was like in my past my class, this is Mr. Boucinha, who was our former teacher, if you remember him.
Hey, Vishnu, nice to see you again, kind of. Gania, yep. So that is the Gania I know. Wonderful. One of my top students as well. Brilliant. oh, I wonder if I should give the adult answer of oh no, that's disrespectful, or if I should give the correct answer of the disrespect. Oh, Vishnu, is your nickname now like Captain Disrespect? Oh no. Oh. Yikes. Well, thank you for tuning in. I appreciate it nonetheless. I think this is actually the first talk that I've ever given live streamed on YouTube.
Let's just give a couple of minutes, just like 2 minutes for people to join.
So one of my colleagues, I presume, is checking in. And he heard the comment that I was referring to Vishnu because he's live streaming while he's in a class. Oh, so he's curious about that. Oh, dear. No.
All right, let's get started.
Yep.
So I'm Siche. I'm the founder and CEO of STEMology. And STEMology is a student-run international nonprofit organization focused on STEM and spreading STEM and its surrounding principles to students across the world. And today we have a live event with Mr. Boucinha, who's a Waterloo graduate, and he's working in the field of synthetic biology. So he's going to share a bit about his work and his past experiences.
I'm Sai Paran, the co-founder and chief operating officer of STEMology. Most of you may know me from school or from past Spirit of Math classes. As Suche said, our organization's goal is to engage students with STEM-related activities all across the world, as STEM careers will be largely in demand within the coming decades. Today, we have our second event with Mr. Boucinha, where he will discuss about synthetic biology. A lot of you may know him as he was your former past Spirit of Math teacher. Our event today will be similar to our first event, where Mr. B will conduct a brief presentation, and then we will have a Q&A session where you guys can ask any questions. After, we will be hosting a trivia in which we ask questions related to Mr. B's presentation. We will inform further details regarding the trivia contest rules and prizing information at the end of Mr. B's presentation. Now, me and Suche will introduce Mr. Boucinha regarding his background studies and current career stances.
So Mr. Boucinha is a secondary teacher licensed with the Ontario College of Teachers, and he's receiving his Master of Teaching from the Ontario Institute for Studies in Education earlier this April. Mr. B obtained his Bachelor's of Applied Science in Nanotechnology Engineering at the University of Waterloo, where he had many diverse experiences from 2013 to 2018.
In the middle of this period, Mr. Boucinha spent 8 months at the Victor Chang Cardiac Research Institute at the University of New South Wales in Sydney, Australia, working on a collaborative project with Oxford University and the University of Ashoka to develop an intensive system for building and characterizing a synthetical bacterial flagellar motor.
The following year, Mr. B became involved with Waterloo's International Genetically Engineering Machine, or iGEM group, to develop a system for the prevention and treatment of neurodegenerative disorders, or NDDs, through the identification of prions in yeast.
Mr. Boucinha has continued his work through his work with the Canadian Synthetical Biology Education Research Group, known as CISBERG, where he and his former iGEM colleagues apply their understanding of synthetic biology and problem-based learning to support the creation of student-driven projects at the secondary and undergraduate levels.
Mr. B currently aspires to eventually become a principal of his own secondary school and to pursue a doctorate in higher education. And now we can hand it over to Mr. B, who's gonna just talk briefly about his work and his experiences.
Thank you so much, Suche and Sai, and good morning, everyone. Thank you so much for having me. It's a pleasure to be here with STEMology, and really appreciate them offering me this platform to talk to you all about synthetic biology today. And specifically, I would like to extend my thanks to my former students who helped to get this organized, Sai and Dira Delash. And also to all of you, my former students who are attending this talk today. You know, it warms my heart to know that I made a great impact on you and that you're continuing to seek wisdom for me. It's truly humbling. So thank you so much for that. And I hope through my presentation today that I can continue to inspire your learning, specifically with something different than math, synthetic biology. So before we get started today, and I'll also share my screen here so that you can all see my presentation. That would probably help. Okay, and if I could just get a thumbs up from the organizers to make sure that that's working all right.
Yep, we can see it.
Yeah, perfect. Thank you so much. All right, so I'd like to begin with with an important land acknowledgement. Specifically by acknowledging the Mississaugas of the New Credit First Nation, whose land I'm learning, researching, and teaching from today, and for the past several years. And while we gather here today on a virtual platform, I'd like to take a moment to acknowledge the importance of the treaties associated with this land that we all call home. We do this to reaffirm our ongoing commitment and responsibility in improving relationships between us and Indigenous peoples of Canada, as well as to sustain their welfare for the coming future. We also do this to improve our own understanding of local Indigenous peoples and their sacred cultures and traditions. Across Turtle Island, we acknowledge the ancestral and unceded territory of the Inuit, Métis, and First Nation peoples who call this nation home. Please join me in a moment of reflection as we acknowledge the harms and wrongdoings of the past and to consider how we can each in our own way move forward in a spirit of reconciliation and collaboration. Thank you. Now, to start off my presentation for today, it's really important to start off with a definition of synthetic biology, because while this is a field that has many, many years of research and questioning and major breakthroughs, the definition alone is still something that's rigorously debated throughout academia and industry today. And so it's super important for us to have a vigorous understanding of what it means from multiple perspectives before we discuss applications and plans for its future. And what we'll do is we'll start by first looking at synthetic biology from an etymological lens, or by looking at the meanings of the words that compose it, in other words. We'll then look at how synthetic biology has been typically represented across various professional and academic spheres, and even through media and Hollywood as well. We'll then conclude with looking at the rich history of synthetic biology across its many years, and investigate those, followed by a discussion of synthetic biology's current place in society and academic research, along with what the future may potentially hold for this really exciting field. Now, to start off, looking at it from an etymological lens, the term etymology, in case some of you haven't heard of this before, is the study of looking into a word or phrase under study from its historical roots. So sometimes in a dictionary, you may see a Latin root or a Greek root, because Latin and Greek languages have many roots for modern words today. Specifically, if we start off with the word synthetic in synthetic biology, we see that this comes from the Greek root synthethenai, meaning to put together and pertaining to. to follow biology, and this may be more familiar to you all who are in secondary school and who are pursuing this branch of scientific study, this comes from two Greek roots, bios, which means life, and logos, which means study. So the study of life, right? Just like chemistry is the study of chemicals, right? And together, we have that synthetic biology is the study of life pertaining to putting life together. Okay, so that's a very basic definition that we'll start off with for this morning. Beyond here, looking at the connotations of the field, you may have before this presentation, knowing the topic that you're entering, you may have tried to search up synthetic biology on an information hub like Wikipedia, and I don't blame you, right? I would totally do the same. But it's really important to recognize that these websites don't take into account the multiple fields and interests that go into synthetic biology, because as I mentioned, this is a really rich field, and it's really difficult to capture the essence of this field from one information hub alone. And some of the things that are missing that I'll talk more about in the later part of my presentation include social sciences, policy development, stakeholder and community engagement, and a plethora of others that you don't normally see on these sites. Now, I'll be also talking about more of the transdisciplinarity of synthetic biology and how my research group is looking at filling this gap through a pedagogical driven effort. But for now, I'd like to introduce a helpful definition from the 2018 Canadian SynBio Conference. And this is the definition here, where we have the design and construction of new biological parts, devices, and systems, the redesign of existing natural biological systems for useful purposes, and the endeavor to make biology and its principles accessible to engineers encompasses the field of synthetic biology. And, when I first read this definition from the conference, I found it to be riveting, not only because of its eloquent prose, but also because it really helps to identify some key implications, right? That synthetic biology is comprised of parts and systems to design frameworks that are accessible to engineers. So the last part with accessibility is especially important because it not only touches on how it's important for a design aspect of things, but that it should be available to as many professionals as possible who want to engage. And that's something that's really at the philosophy of my research today, is making sure that, I can engage with groups like STEMology and help everybody to learn more about this field. And I'll be jumping back to this idea multiple times in my presentation, as you'll see. Now, looking at things from a more historical lens, right, it's really important to note that SynBio wasn't always incredibly clear to us, right? Like many other scientific fields, there's many mysteries that enshroud the field and that have, in some cases, continued to plague the field today, but that just makes it more exciting to try to learn more about it. For those of you who are familiar, for example, with how electricity was discovered and researched, initially individuals thought that lightning was, from the gods, right? They thought that lightning was, some supernatural and mythical phenomenon, until they realized that, oh, this is just the movement of these little tiny particles that even today still befuddle us at times, called electrons, right? So knowledge evolves, right, over time. And synthetic biology is no different. So as with all of our ventures that we've gone on with science, there's still many fundamental discoveries that are found, right, with every few years. And this really helps to drive the passion for researchers all around the world with this field. So to look at some sort of a timeline, these are some events that I found to be really helpful for understanding the field. I just wanted to identify some really important phases of synthetic biology. So the first here is molecular biology. Okay. And molecular biology gained prominence in the 1950s when the structure of double helix DNA and genetic heredity were established. So for those of you who have taken grade 11 and or grade 12 biology, you'll be familiar with Mendelian heredity, right? So the idea that if you take a Punnett square, you cross characteristics of two organisms, you'll get an organism that shares genes from both parents, right? And the DNA structure is essential for making sure that we can pack as much information as possible into a very tiny volume, right? The double helix DNA is certainly made to have that high information density. And as you might imagine, because these are so fundamental to life, a lot of science, a lot of fundamental science at the molecular level was done during this period of time. But skipping over to several years down the line into the 1970s, we have molecular biotechnology. And at this point, we've had the development of plasmids, rings of DNA from prokaryotes. So if you're familiar, and this is kind of going back to grade 10 biology with cell theory, you have prokaryotes and eukaryotes, right? Ring DNA, very simple, right? No membrane-bound organelles, just a very simple organism. But then you also have our modern multicellular organisms that have nuclei, they have the mitochondria, they have the Golgi apparatus, right? They have all these organelles that help different functions to happen. But we found that plasmids were really nifty for modifying because it's just one giant ring of a sequence of code, basically, right? It's a lot easier than taking our modern DNA, unwinding it, and working with it from there. Even though, as I'm sure you're familiar with what's on the media today, this is something that we're becoming better and better at doing. But regardless, back in the 1970s, this was huge, being able to modify these rings of DNA. And when we were able to integrate some genome work in here, we were able to see many applications in the pharma and agriculture industries where we could develop new remedies for different diseases and where we could help crops to build more defenses against different biological organisms that would try to harm them. And this has certainly helped humanity to be sustained over time because it keeps us more healthy. And a lot of this science, given these applications, involved more applied science. So now we don't only have fundamental science that's being worked on, but we now have applied science at a couple of levels, the molecular level and the systems level. So kind of looking more outwards to an entire system or an entire setup. The last one would now be synthetic biology, where we have, beginning in the 2000s, a huge development into DNA synthesis, genetic circuits, and the redesigning of a phage genome, to name a few. These are certainly not the only ones that exist, but they were some of the most notable. And we also noticed a rapid extension in academic labs and iGEM, which I'll talk about more in a little bit from my experiences. And these just help to get a lot of a spotlight on the field, on the international stage. More universities and more governmental institutions started funneling money into the field. And now it's definitely something that does take up a huge chunk of a government and an institution's budget if they have a stake in synthetic biology. And as you might imagine, this also imagines a lot of applied science at the molecular and system levels, because now we have all the fundamental tools that we need. And now it just comes down to refining and making better stuff, right? So beyond those discussions of phases, though, I would also like to talk about some notable events. And these are really important for understanding where we are right now, just kind of envisioning where did we start way back when, where are we right now? And where this starts is with 1950, with the structure of DNA found by Watson, Crick, and Franklin. So that's that double helix structure that you may have seen the X-ray crystallography picture of, where it's just like a shadowy X figure. But that was so like legendary, right? For getting this to where we are now. That was the start. From understanding the structure, Jacob and Minot in 1960 then developed something called the operon structure. So in case any of you who are in grade 12 are familiar with this, this is basically the part of DNA that's responsible for inhibiting and promoting certain processes. So let's say if you consume a dairy product, your body will say, oh, we need to produce lactase, right? The enzyme that helps to break down the lactose in milk. But if there's no milk present, then your body isn't going to produce that because that would be a waste of time and energy. So understanding the operon structure was really important for us to understand how our bodies think at the genetic level and how they're able to regulate themselves using this special code. Beyond that point, Smith and Wilcox in 1970 found these really impressive parts of DNA called restriction enzymes. And the way that I want you to think of restriction enzymes is imagine a piece of tape that you use to, let's say, connect two pieces of straw together. You could imagine that the straw is your DNA sequence. And what Smith and Wilcox found is that at these particular points where you have the tape, you could actually unstick them in order to remove a sequence of DNA and then insert a completely new one that has these sticky ends. So you could imagine that this is really critical for modifying a plasmid. If I can remove something and then put in something else that has the same ends, then the DNA will be read in the same way, but it'll produce different things because now it has different instructions. So restriction enzymes were some of the first modification techniques that were developed for us to, break through and actually modify plasmids that we had available. And in 1977, Sanger was a fellow who was able to sequence Phi X174, which is a specific type of plasmid. And this was one of the first sequences that we had for DNA. So actually knowing the order of GCAT, right? The 4 nucleotide bases of DNA. So he was actually able to go through it and actually tell, oh yeah, here's the entire sequence in a text file. That's the organism's DNA. Right. So that's pretty interesting, right? And it was certainly something that was fundamental to a lot of our modern DNA sequencing techniques. And in 1985, Mullis topped up this work by developing something called the polymerase chain reaction. And in case you're unfamiliar with what the polymerase chain reaction is, this is really something that you may have seen in the news with COVID. It's a method that some researchers can use to replicate the proteins that you see with the coronavirus. So you can easily tell if somebody's infected, because if you drop one of those sequences in your machine and you replicate it 1,000,000-fold over, you should certainly see it in the sample that comes out. And if somebody tests negative, you shouldn't see anything produced from that reaction. Now, there are some issues with using it for that purpose, but I could definitely tell you that polymerase chain reaction is definitely something that we've had a chance to use even in laboratories today in order to build a lot of DNA all at once. Now, a more modern version of DNA assembly is the Gibson assembly method developed in 2009. And this is a really special cloning method that takes smaller DNA fragments and basically uses a special constant temperature process to bind them into one longer single DNA structure. So definitely a really interesting thing, although it is proprietary. So there's very limited information on that I have to share with you at this point. But there's definitely some really interesting things that are happening there in terms of the biochemical aspect and using, thermal physics in order to actually get those that assembly to happen. And one of the most recent things that was really notable for me in my research group is something called the toggle switch. And this is a really interesting part of synthetic biology for microbes specifically, because it gave researchers a way to try to program a kill switch into microbes that wouldn't require the use of harmful chemicals, right? So instead of, let's say, using alcohol or bleach to kill off multiple microbes, they were actually able to code something into the DNA of these microbes that said, if such conditions exist, terminate all processes, like stop taking nutrients, stop cycling water, stop getting rid of waste, just shut down completely. And this was a really interesting breakthrough because you're now able to tell a microbe that, okay, under these conditions, do these things, but under other conditions, this is super dangerous, shut everything down, right? So really, really profound for even disabling parts of microbes that could harm us. Right? If you modify one microbe and you have it multiply and you have it take over a whole colony, that's your ticket, right? To be able to influence control. So definitely one of the most profound examples of exerting control on a species like different microbes through the kill switch. And definitely an important part of a lot of experimental design today. Now, reworking our previous definition after all of that investigation into history, into the wordplay, and into connotations, it's really important that we make a revised definition. And this is one that one of my colleagues proposed. And he mentioned that synthetic biology is both a fundamental and applied science that can be wielded to explore truths about life and to design it for the betterment of human societies and the environment. So I really like that part, especially where he talks about exploring truths, right? So recognizing that there are multiple aspects of this that are really interesting to think about, not just the science part, right? Like not just the fundamentals, but also how it can be used to help improve our world, right? Because to ignore the implications of such a science would be a disservice to everyone, because you have now something really cool that works in a lab that stops there, right? And it's really important for us to think about, what does that product do? What does it imply for our world? And so a new contemporary definition of symbio can be realized from everything that we've talked about so far. And in reflecting on that implication piece, we can now see that symbio is an interdisciplinary field that pertains to the creation of, you could call them living robots, right? So if you visualize some molecular and cellular machinery that could be designed de novo or brand new from characterized parts and systems for a well-defined task or a set of tasks. And in doing so can lead to new fundamental discoveries in molecular systems biology. So it kind of loops. The fundamentals inform the new things, and the new things can inform the fundamentals, right? And it's a cycle or a spiral of knowledge, if you imagine it that way, that really helps us to grasp this field better and use it to our advantage. And this is also something that my brilliant colleague talked about. And, I'll be mentioning his name quite a few times today. He was a pretty big inspiration for me personally. Now, before I go into some of those points, I think it's really important to, again, remind you that everything up to this point is traditional, right? So everything that we've been looking at so far is through a mostly traditional lens, maybe minus the part of the implications, because that's still something that's being worked on in our modern decade, or for the past couple decades. And it's really important to think about, where do we go from here, right? So thinking about that purpose of synbio in our contemporary world. Now, one of my biggest influences when it came to synthetic biology was through the International Genetically Engineered Machine, or iGEM competition. And this is the world's first student design competition that's focused on synthetic biology and includes high school, undergrad, and graduate teams. It was formed by the Massachusetts Institute of Technology, or MIT, as I'm sure you're all familiar with that name, in 2003. And it still runs to this day with its giant jamboree occurring in the fall season. Last year was the first year that we ever did a virtual conference. And, this year we're going to be sticking virtual as well. But it's anticipated by 2022 that we'll go back to our current location of Paris, France. So a pretty big event that hosts a lot of teams from around the world. And this is where I began my SynBio journey. Back at the University of Waterloo's iGEM team for the 2016 and 2017 competition years. And it was arguably, and I firmly believe this too, the first major purposeful implementation of synthetic biology for the general public, because even now you could go back to previous competitions and view projects through their websites and actually understand the work that they're doing. So there's definitely a huge engagement with the general public. That's a really big focus of this competition. And for that, I definitely see it as being more applicable to even the most wondrous thing occurring in a lab that doesn't make it to the market, or that doesn't make it to the general public's eyes. The impact factor is just so much bigger with iGEM, in my view. And even to this day, through the COVID-19 pandemic, I had a chance to judge for the competition last year, and I got to saw some amazing work from various teams around the world. And it's clear that even a pandemic isn't enough to stunt the growth of this competition and a lot of the creativity that exists. And Patrick Deep was one of the people that I met through this competition, and he's by far one of the most influential people that I've ever met. And you've seen his name mentioned before in my presentation, and you'll see it mentioned again for a good reason, because he's the one that really helped me get started on the major path of my academic and professional life. Before I met him, I was kind of confused as to what I wanted to do, but he really helped me to get a lot of those answers filled out there. And back in Waterloo, he ended up graduating a year before me to move on to Toronto and pursue a PhD in biotechnology there. And it was sad because at that point, there was about a couple and a half years of radio silence since, I ended up getting really busy myself. I ended up just kind of leaving social media behind, suspending my Facebook account, and just kind of focusing on, getting a lot of work done for my teaching career. But, flip to a couple of years after that, I don't know why I went back to Facebook, but I did. I reinstated my account, went through my friends list, went through old convos, and I came across Patrick's profile. And, I remember some fun times, right? Things were fun, things were really engaging. So I sent him a message, just to say, hey, I hope we can meet up again, right? And this was back in August of 2019, just before my teaching career started in Teachers College. And, I thought this probably isn't going to go anywhere. It's just a simple hello, right? No big deal. But to my surprise, Patrick actually wanted to meet with me after all these years. So we thought to meet up at Famoso, which, by the way, if you've never gone to Famoso, really great place for Neapolitan pizza. This is actually a really special kind of pizza where you actually have to eat it like you're folding a wallet, right? So you actually have to fold it and then eat it like that, because it will fall apart if you try to eat it the Western style. And by Western, I mean like American, right? So don't make the same mistake that I did when I ate it for the first time. Right. And, I've always known Patrick to be a great passion seeker, somebody who's incredibly driven and whose charisma really inspires many people to follow his lead to success. And, ever since I met him, I found that strong aura emanating from him. And even on that evening, I could tell he wanted to tell me something. I could see it in his eyes. I could see it on his face. There was something he really wanted to tell me. And sure enough, this was about his latest passion project, the Canadian Synthetic Biology Educational Research Group, or CSBIRG, right? And through this initiative, Patrick wanted to follow one of his greatest passions in synthetic biology, to improve access to education in this field within Canada and to enhance the quality of this education overall. And the reason why he reached out to me was because he knew that I was studying pedagogy, or the study of learning at the time, through OISE. And he really wanted me on board to help advise CSBIRG members on best teaching practices for research and, for future projects, because, education is in the name of the organization. So he thought, hey, somebody from education would be perfect for our group. And even though CSBIRG started off with just a small room of less than a dozen members, we've now doubled our numbers over the course of the pandemic and have officially incorporated as a nonprofit organization just a few weeks ago. And some of our past research was actually our first paper in the Canadian Journal of Microbiology, which got fully accepted at the end of April. And this research, in case you're ever interested to search it up, excuse me, this research really helped to focus on the Canadian iGEM landscape, including team dynamics, institutional support for student teams, and synthetic biology education access. So some really interesting pieces there. Really encourage you to check it out in case you're interested about this side of synthetic biology. Especially for me, it was definitely a personal achievement because this is my first paper of primary authorship. So, you can bet that once the pandemic rolls out and finishes, I'll be printing this out on some super nice paper, some super nice leather binding, and I'm going to put that on my shelf immediately because, you got to do that for your first ever paper. So. Now, what's happening right now with our team, right? So we're definitely not stopping there, and we're certainly not stopping as we're going along. We have a lot of projects on our plate right now. One of the biggest ones was iGEM team mentorship across the country. And we're also currently running a project with an Ontario private school. I could only mention so many details now since it's still in the planning phase, but if you keep tuned to our social media channels, which I'll have at the end here, you'll be able to learn more. But something that I can talk about are the courses that we've offered online over the past few months of the pandemic. And these are SINBI 1 and SINBI 2. So SINBI 1 is a course that's focused on developing technical skills, so wet lab and dry lab skills in order for students to better support their own iGEM teams at home. And SINBI 2 is designed to help people who are understanding of the technical aspects to understand the non-STEM aspects of SynBio. So looking at multiple components of implementation sciences, that includes discussions of ethics, business, marketing, government regulation, and stakeholder engagement. So the second course is kind of looking more into that implication side that I kept harping to in my presentation. And, sadly, there are a couple dimensions of SynBio that aren't considered as strongly these days. The social, ethical, and global implications, and also the financial and commercial implications as well. And one big thing that, at CSberg that we're trying to do is we're really trying to contribute to the new face of synthetic biology by committing to education that integrates policy development, governance, and partnership with the public to really create products and services that are accessible and beneficial to all. And this vision for the future of SynBio needs to be done by moving synthetic biology from an interdisciplinary discipline, where only STEM concepts are considered, to a more transdisciplinary field that welcomes perspectives from all other professional domains. And a great example that I show to people who are trying to think about what this looks like is with my own home field of synthetic of nanotechnology engineering, And you can see by this diagram here that I still refer to very often. I think it's very well done. It's not my own work. You can follow the link there to that diagram and its author when you have the time. But I thought that it painted a really brilliant picture of nanotechnology from a transdisciplinary lens. And what you'll see is that you start off in the multidisciplinary phase with physics, chemistry, engineering, biology. And over time, they merge to form their own subfields, right? Like biochem and mechanical engineering, depending on which ones you're combining together. And eventually, these will all coalesce into one big field, nanotech. And the thing with nanotech is that you can't have nano without also having other fields as well. Because with nanotechnology, there's a lot of different complexities. There's discussions of ethics that need to be considered and business. A lot of nanotechnology ideas are in the startup phase. So you have to need to know how to run a business. There's a lot of discussions of law, right? What is legal to implement and where and under what conditions? And also computer science, because sometimes it's really expensive to work with these things. So if you can code it first, that would be preferred, right? So you can see how nanotechnology really does rely on pulling on a lot of other things in order for it to be a viable field. And if you need a way to think of transdisciplinarity, you could think of it as transcending boundaries or silos that really separate different fields from each other. And it's in our group's core creed that, we believe that SynBio is one of those fields that really needs to undergo a similar revolution in order to gain a more widespread global acceptance. So what does the future hold for CSberg? Well, we do have our eyes set on a couple of partners for the future. The first is University of Toronto Schools, which some of you may be familiar with as a prestigious private school in Toronto. And our team is hoping to get connected with the school in the near future in order to run some workshops and professional programs there centered around SymBio. The second, which is probably more familiar, is the Toronto District School Board. A fun fact, the TDSB is still the largest board in the province, making it also home to the largest number of independent schools. And these are schools that have a certain degree of autonomy from the board, which means that they have the ability to make more independent decisions from the board's decisions. And this would make it a really great set of places to implement our workshops, right? If a school's administration decides that they want to take us on, they don't always need to get the board's permission to do so. And so we hope to do this before developing rapport for general public schools, so schools that aren't independent and they're just regular schools. It is our mission to try to integrate ourselves as much into these systems as we possibly can. So here's the contact info for our group, and I'm sure Suche and Sai will be sharing this with the audience following my presentation, but I definitely encourage you to follow us through Facebook, Twitter, Insta, LinkedIn, whatever you'd prefer, because we do release our most recent information there about where we're at with research and what course offerings we have in case you'd like to enroll in the future once you're in your undergrad stage of study. And feel free to also drop us a note either at the CSberg email or at my professional Gmail account there, and I'd be happy to answer your questions and, happy to hear from you in case you're interested to even join our team. That would be cool too, right? If you feel like you're a good fit. So thank you so much for attending my talk. I really appreciate your time and your attention. And if there's any questions, I'd like to take those now. And in case anybody does have any questions that they'd like to ask vocally, that would be preferred, just because I don't think I could see the chat right now. Oh, okay, there we go. I got the chat open. All right. So in case anybody wants to ask a question in the chat as well, I do have that open and accessible.
All right, then I guess we can get continued with the trivia.
Yeah,
And,
sounds good.
Yep. And Suchi will be sending the link in the chat where you guys can just access the trivia. You guys can win real money. And underneath the trivia, beneath the Google form, there's a bunch of rules that explain how the awards will be given. So there are 3 easy questions that are each worth $5, and then 2 medium questions that are each worth $10. And Suchi, you could explain the hard question.
All right, the hard question will be given orally by Mr. B, and there will be a diagram on the screen relating to it. So yeah, that's after the trivia for the easy and medium questions.
Mr. B, I think Chrissy asked a question. How can we integrate these concepts in the business field that were discussed in your presentation?
Perfect. Yeah, that's a great question, Chrissy. Thank you for asking. So in terms of implementing in the business field, definitely on my team, I'm not best qualified to answer, since one of my friends, Colin Pierce, he's really more of the fellow to ask for business implementation. But, from my discussions with them, and also from, helping out from the pedagogical side of things, it's really important to recognize that, with synthetic biology, just like nanotech, you have a lot of people who have some really great ideas and who want to bring that to the market, right? And this could even be at the higher level too. Like if a university professor, let's say with their lab team, discovers something that could be revolutionary for an industry, they need to go through several different exercises when it comes to bringing that to the market. And in terms of looking at a business analysis, it involves really understanding what your market base looks like, who's going to be buying into your product, right? What kind of business model are you going to have? Are you going to just have like a simple licensing setup, or are you going to have somebody else like deal with it for you and you just supply the science, and you go through like a distributor or a special publisher for it, or do you sell out completely, right, and kind of appraise your idea and sell it to a bigger company that can handle it, right, whether it's the government or private company like GlaxoSmithKline, for example, or Thermo Fisher, Roche, right, one of those big names. And also just, if you do decide to go it on your own, right, and you decide to really sell this idea, right, like you need to understand that, business operates differently depending on which country you're in, right? Things like intellectual property will be something that you need to look into, right? How can you best protect your idea? Do you use patents? Do you just use copyright protection? And what do you need to do in order to maintain that protection for as long as you can? Certainly with Canada, we have our own set of copyright laws, but the US also has their own. So depending if you want to go beyond the border, you really have to do your research. So definitely a lot of components that would take a lecture of their own, I assure you. But if you'd like to ask any more questions about those details, I highly recommend reaching out to the CSberg email and just mention Colin Pierce, and I'll put his name in the chat here just to spell it out for you. And he's a great colleague of mine to address those kinds of questions too. Any other questions?
I don't see any hands up. So, all right, guys, you guys can fill out the Google form for the trivia right now, and then once everyone's done, Suchi will inform, or Mr. Suchi will ask the hard question, and then Mr. B will answer it or correct you guys based on his judgment. Mr. Boucinha, I have a quick question. It's just a general question. So, if you were to give advice for students who are graduating from high school and going to undergrad programs in post-secondary institutions such as universities, do you have any advice for high school students?
That's a great question. And I mean, I used to, of course, be a high school student myself. So I'm kind of just imagining if I could talk to my past self, what would I say? Um, Probably one of the bigger things, since SynBio is still quite general, and it's one of those fields that I'm pretty aligned with, given that nano and SynBio both have that flair. Realistically, you could still continue to pursue your passions in STEM and get involved with this field, even if it's not in STEM, right? Like, let's say you wanted to go more into policy development, you could absolutely join an iGEM team and they'd be happy to have you. Like, honestly, pursuing your passions, I would say, should come first and foremost, just because your passions could take you anywhere. From what I've seen, especially, that's definitely true. As a small anecdote, I had a colleague, a friend of mine back in Australia, back when I was studying there. He transferred from the private sector doing design on places like CAD and Photoshop and whatnot, and he ended up successfully getting a PhD in biotech. So, it's and he was at the age of like 35, right? So, you could always change your mind and refine your thoughts on where you want to go, and that's totally fine. But what comes first and foremost is that passion piece, right? Like, what do you want to do? Where do you want to go? And from there, the pieces fall into place. Just keeping your eyes and ears open, right, for new opportunities. I would say that's probably one of the biggest things, is just making sure that you're open, right, for whatever comes your way.
Sucha, could you just inform us how many more students are yet to fill out the forms so we can get the hard question answer? Or
All right, 2 more students still have to do it.
okay.
All right, I think we can get started with the hard question now.
All right. So I guess Suche could ask it.
All right. Just split up, and I have to share the diagram also. So right, here is the diagram. I'm gonna share my screen and the diagram will show up. All right, do you see the diagram? It's,
Oh, yeah, we can see it.
all right, so the question is, using the four set Venn diagram that Mr. B presented earlier in his presentation as a guide, construct a four set Venn diagram to show how synthetic biology was constructed from four initial multidisciplinary fields. The complete answer will also identify what fields make up the smaller overlaps. For example, physics and chemistry make physical chemistry, physics and engineering make mechanical engineering, physics and physical chemistry plus mechanical engineering make mechanochemistry. So we're looking for a creative answer with this submission rather than technical correctness.
So you guys can just raise up your hands, and then whoever raises their hands up first can give it a shot. This question is worth $15. So I suggest you give a shot at it and answer it.
I think we have a request in the chat just to repeat the question there.
Yep, I'll just put it in the chat right now.
Yeah, perfect.
Here, let me just share the screen with the question. It's a pretty long question. So.
Oh, yeah, there we go.
Posted it in the chat.
Perfect.
Sorry.
Okay, I see Sarah's hand raised.
Oh, sorry. I think that was by mistake.
Oh, okay. Okay. False alarm.
Any answers, anybody?
Anyone can give it a free shot. We'd really appreciate it.
I mean, at this point, I'm thinking that you can have multiple tries also. We don't have that many participants either, so.
Yeah.
Kind of wondering how I should judge quality here.
If anyone wants to type it, here's a link to the document in the chat. It's an empty shared document where you can type your answer.
Suche, is this did you enable edit access for the doc?
Yes, I did. I did.
Okay.
Um, I should be able to edit.
Also, Suche, Ganya told me that she has some Wi-Fi issues, so that's why she left the Zoom. But I'm in contact with her. I'm asking her the hard question, so I'll just tell you her input, all right? And then Mr. Boucinha can say something.
I mean, really, guys, just take a shot at the question. It's okay if you're wrong.
Okay, so Chrissy answered. I guess Suchi, you could read it out loud if you'd like.
It's in the chat, so. I think it would be easier if Mr. B just judged it.
Or yeah, I guess Mr. B could just read it out.
Sure. Yeah. It's a good start. It's a good start. No. Just kind of wondering what those overlaps would specifically look like for SynBio. And I suppose bio and chem is one, so biochem, right? And then maybe thinking about some other overlaps as well. If I can maybe make a recommendation in case anybody's struggling particularly with this question, maybe start off with 4 fields and try to imagine what those overlaps look like.
Mr. B, I think Chrissy put a couple of options like in the chat. For example, biology and engineering produces the new undergrad program of bioengineering. And then that can also branch out to like medicine to give you guys a couple of ideas. And yeah, isn't that what like you're looking for, Mr. B?
Yep, it is.
Okay.
And I'm especially curious. So that's like kind of like what I would call the first overlap. And then if you kind of imagine what happens when you bind those together, what do you get from there? Right? Okay. So now maybe try to imagine what happens when you take bioengineering and you combine it with physical chemistry. What would that give you? And I know the example identifies medical health sciences, but what else can you think of?
Mr. B, I think there is some more answers in the chat.
Yep. Okay, yep. So Chrissy has the right idea there. So that's the ones that form nano. What would be the 4 kind of deeply integrated fields that would make up SynBio? That's what the question's asking. So if you could imagine 4 big complex pieces that unite together and make SynBio, what would you identify? I think at that point I'll give like a thumbs up to the answer that I get for that. Because making a Venn diagram virtually is difficult. I'm sure Sai and Vishnu, you could agree with that.
Yep, of course.
4 set, 7 set Venn diagram.
And then remember how we discussed about unions and then
unions, intersections, complements. Yep, I remember. So Suche, I'll just respond to your question vocally there. In a way, yes. However, I'm kind of also looking for like the transdisciplinary aspect, right? So the inclusion of biology is of course necessary, but what about some other fields? Right? I want to make sure that my audience is considering that important factor there.
Mr. B, so your question is just basically asking how each of those four, or any four major fields link together to form SynBio, right?
Right.
Okay, so could I give it a shot just for, the sake of my knowledge?
Sure, yeah, absolutely.
Yep, and what I'm guessing is that biology and chemistry, they both like intercalate to form biochemistry, right? So in that sense, we're just discussing about how molecules in living organisms interact within each other. In the case of SynBio, we're gonna I guess that correlates in the sense that we're studying the way how molecules or cells interact amongst each other during a chemical reaction, such as growth, cellular growth, and cellular respiration. And I guess would that kind of work in this, like for one of the points?
Yep, I would say so. Absolutely. And even looking over to these answers, I think so far, Chrissy identifies like the most complete answer for this one, I would say, especially with the inclusion of genetic engineering there.
Ganya told me she's gonna give it a shot after some time right now.
Sure.
In the meeting in, so I guess we'll just wait for a bit.
Sure. Yeah. I mean, yeah, we could also I could also let you know my decision following here, and then we could just contact the winner from there.
I give Ganya the link to the doc, and all the later answers could be written down there, and I can share the doc with Mr. B.
Sure. Yeah, that'd be perfect. Yeah.
Okay, sure. Suchi, can you send the doc to me?
I think the link is in the chat.
Okay.
Yeah, I do apologize as well, because I did think that we would be done by around now, just because at 12:30 I do have a meeting that I need to bounce into. But yeah, I mean, I'm happy to look over any answers that come in though following our session. Cool.
Right then, the doc is in the chat. If anyone wants to put down an answer, I'd say I'll give you like about 24 hours maybe, and if you want to give it in by then, Mr. B can rate it and decide which one's the best answer. All right, so thanks everyone for coming to the event today. It was great to have you all.
Thank you very much, guys, for coming here. If you have any questions for